So, as we have been talking about again and again, if there is biosynthesis, there is also degradation. If a compound is formed by the agency of energy, by the utilization of energy, it also gets degraded and you get energy out of this. If you see the phospholipids, it has certain bonds. For instance, the bonds which attach the glycerol moiety of the phospholipids with the fatty acid or with the phosphate moiety. So, and their breakage is important for the degradation of the molecule, degradation of the phospholipid. Important, I mean, enzymes which are involved in this breakage of these bonds are known as the phospholipases. For example, here you can see A1, A2. These are the important lipases which are involved in the breakage of the bonds which are connecting the fatty acid moiety with the glycerol moiety of the phospholipid. And similarly, there is lipases, the D or C lipases, which are involved in the breakage of the phosphate moiety from the rest of the phospholipid molecule. So, one of the example of this is phospholipase A1 or A2. They give rise to a compound which lacks a fatty acid. So, the compound contains one fatty acid and one phosphate moiety, and that is known as the lysophospholipid, and the enzymes which act upon them are the lysophospholipases. One of the products of this action of the lysophospholipases is, in addition to the rest of the compounds you also get some intermediate compounds specifically which play very important role in signaling pathways. They act as messenger molecules or signaling molecules like diacylglycerol or IP3, and we will talk about them in detail when we will be talking about the cellular signaling pathways which are involved in different which carry out, or because of which different biochemical pathways are regulated. Arachidonic acid is also an example of same sort of messenger molecules. Another very important, I mean, part of lipid metabolism or phospholipid metabolism specifically is that it's not all about degradation to the final constituents. Also, sometimes remodeling also occurs. So, as a result of remodeling, you get some new molecules which your body needs. For instance, here is a general phosphoglyceride, and it's acted upon by the phospholipase enzyme, phospholipase A2. So, as a result of this, the fatty acid R2 moiety is released, and you are ended up with the 2-lysophosphoglyceride. Now, this 2-lysophosphoglyceride, the R group which is lost here, it can be replaced by R group from another compound. As a result of this, you get a new R group here, and this new R group is different from the R group which is replaced, and you get a new compound. So, this is known as the phospholipid remodeling. As a result of remodeling, you can get a compound which is needed in new circumstances in your body. But depending upon the circumstances, depending upon the need, this sort of remodeling occurs through the action of lipase. It simply replaces one R group with another, one fatty acyl group with another of the phospholipid, specifically the phosphoglycerides. One of the implication of this, because this is a very important process, it is because it is done to fulfill the needs of specific tissues. For instance, cardiolipin, if it is it doesn't undergo real, actually correct remodeling, what will happen that it will give rise to birth syndrome, a very rare but very dangerous disease in which your heart size gets increased. So, in this way, you can see by sometimes even simplest of phenomena like remodeling of compound in which only one part of the compound is replaced by another compound, major portion of the compound remains the same, only one portion is changed with other, and it can give rise to defect in this process, can give rise to some really lethal consequences.